Normally, right, when we look at some sort of complex, highly functional system, say a car engine or, I don't know, a global computer network.
Right, or even like a massive suspension bridge.
Exactly. We tend to approach it with this expectation of engineered precision. Like if you plug a diagnostic machine into a car, the screen just spits out a very specific error code.
Yeah, the mechanic points to a cracked spark plug, and that's it.
Right. The system is basically binary. It's either functioning or it's broken. And it operates within this totally closed loop of, predictable physics.
And we find that predictability really comforting
Oh, absolutely.
because the human mind is just naturally drawn to categorization.
Yeah.
We want visible, deterministic causality. We want to believe that if we just understand the blueprint, we can perfectly predict what the machine is going to do under literally any circumstance.
But the thing is, the moment you step out of the garage and into the natural world specifically to study animal behavior, that comforting diagnostic machine just completely shatters.
It's gone.
Yeah. We're no longer looking at clean blueprints. We are looking at a behavioral landscape that is, frankly, profoundly murky.
Oh, the absolute definition of diagnostic muddy waters.
Right. I mean, you observe a bird executing this incredibly elaborate, exhausting dance for an audience of one, or you watch a ground squirrel actively risk its own life just screeching at the top of its lungs to alert its colony that a hawk is passing by.
Yeah, and there's no simple binary error code to explain those actions.
Not at all. Which is exactly why you're here with us today. As an undergraduate student diving into the fascinating world of animal science, you need a shortcut to understanding this stuff. So welcome to the deep dive.
It's great to be doing this.
Yeah.
Because unlike a carefully engineered car engine, an animal is the product of hundreds of millions of years of just messy, chaotic, brilliant evolutionary compromises.
Legacy code, basically.
Exactly. Every creature alive today is just a patchwork of biological legacy code.
Which brings us to the core of our mission today. We're pulling from an incredible stack of source material for this deep dive.
Yeah, we've got classical ethological texts by Mark Ridley and Eric Cassell, comprehensive textbooks on behavioral ecology.
Some really heavy modern neurophysiological research, too.
Right, alongside the observational insights of researchers like Jeffrey Masson and Lee Allen DeGotkin.
And our overarching goal here is to explore the fundamental mechanisms behind, why animals do what they actually do. We're going to treat animals not just as a collection of cool creatures, but as highly complex living algorithms.
Algorithms that have been relentlessly shaped, compiled, and debugged by the unforgiving pressures of natural selection.
Oh, I love that phrasing.
Well, it's true. To truly grasp the subject, we have to recognize that an animal's behavior is just as much of a biological adaptation as, say, the sharpness of a predator's tooth.
Or the thermal insulation of a mammal's coat.
Exactly.
So we're going to trace the origins of that behavior from the absolute microscopic baseline. We're talking coiled strands of DNA and migrating embryonic cells.
Building a nervous system from scratch.
Right. All the way up through individual learning and finally observing how those behaviors manifest out in the complex, high-stakes environments of predator-prey arms races.
And primate societies.
Yeah. But to do that, okay, let's unpack this. We have to start with the foundational engine of all biological science. Natural selection.
Right. So modern evolutionary theory rests on what we can essentially call two twin pillars.
Okay, weigh them out for us.
The first pillar is random genetic mutation. These are the spontaneous, entirely unpredictable changes in the DNA sequence that happen during reproduction.
So they just inject constant variation into a population.
Exactly. And the second pillar is natural selection itself, which acts as this ruthless filter on that variation. It's generally summarized by the phrase, survival of the fittest.
Right, and the word fittest is where people always get tripped up, I think.
Oh, constantly.
Because in common parlance, it sounds like we're talking about athletic prowess, like going to the gym.
Yeah, being the most jacked animal in the forest.
Right. But in an evolutionary context, fitness isn't necessarily about being the largest or the strongest or the most aggressive. It's actually a strictly mathematical concept.
It really is.
Fitness is just a measure of survivorship and reproductive success. It's literally just about how many copies of your genes make it into the next generation.
Which is such a vital distinction to make, because if an individual has a random genetic mutation that makes it slightly more cryptic-like, better camouflaged, or slightly more efficient at digesting a specific plant.
Or even just slightly more cautious in a dangerous environment.
Exactly. If that mutation translates into surviving long enough to rear more viable offspring than its peers, nature selects for that trait.
The frequency of that specific genetic variant just increases in the population over time.
Right. And over geological timescales, this relentless sifting process is what generates the immense diversity of the entire biosphere.
And Charles Darwin understood this brilliantly. He explicitly argued that natural selection doesn't just shape physical morphology.
Like the color of a mouse's fur or a finch's beak.
Yeah. He recognized that complex, highly programmed behaviors, which he basically just called instincts at the time, they are subject to the exact same mechanisms of variation, inheritance, and reproduction as any physical organ.
Because you can't separate the structure of the animal from what the animal actually does.
Right.
If a specific behavioral pattern increases an organism's reproductive fitness, the underlying neurobiological architecture that generates that behavior is going to be favored by selection.
And what's really wild to me is how frugal evolution is. It almost never designs a completely new behavior from scratch. It is the ultimate tinkerer.
It works with whatever materials happen to be lying around in the genome.
Yeah. There's this concept in our sources called evolutionary opportunism, and it perfectly encapsulates this idea. Evolution will take a behavior that evolved for one highly specific survival reason, and then over time completely co-opt it for an entirely different purpose.
It highlights the sheer plasticity of biological systems, really.
Yeah.
We see this all the time when selective pressures in an environment shift dramatically.
Right. The sources outline this fantastic hypothetical example involving a ground-dwelling bird that's lost the ability to fly.
Oh, this is a great illustration.
Yeah, so imagine this bird's ancestors were fully capable of flight. But over thousands of generations, maybe there's no ground predators and there's tons of food on the ground.
So the selective pressure to maintain heavy, metabolically expensive flight muscles just disappears.
Right, flying literally becomes a waste of energy, so the species becomes terrestrial. However, the bird still retains the deep neurological wiring, the motor pathways, and the vestigial muscle memory for a pre-flight wing movement.
It physically possesses the circuitry for a behavior that is now ecologically totally useless.
Exactly. But evolutionary opportunism never lets a viable neural circuit go to waste.
No, it doesn't.
So perhaps a male of this flightless species idly performs this ancestral pre-flight wing flutter, and entirely by chance, a female in the population finds this visual display stimulating or attractive.
She preferentially chooses to mate with the male who flutters.
Right. Suddenly, that ancient pre-flight movement is subjected to a completely new set of selective pressures. Sexual selection.
The behavior is locked into the species' repertoire, not because it offers any aerodynamic advantage, but strictly because it's a courtship display now.
It's wild. And the formal ethological term for this process is emancipation, right?
Yes. The behavioral unit is emancipated or freed from its original functional context. It becomes ritualized.
Which feels like a critical warning for anyone, especially you listening as a student when you're studying animal behavior. You cannot automatically assume that the current utility of a behavior provides an accurate historical account of why it originally evolved.
Absolutely not. You have to look deeper.
And to truly visualize how selection manipulates behavior over time, I think the clearest window we have is artificial selection.
Human intervention.
Right. Humans have been playing the role of the evolutionary filter for thousands of years, and analyzing what we've done to domestic animals reveals some pretty incredible insights into genetic linkages. The sources detail a concept called the domestication syndrome.
Driven heavily by the work of biological anthropologist Richard Wrangham.
Yeah, and it fundamentally changes how we view our pets and livestock. Because when humans began domesticating wild species like transforming wolves into modern dogs or wild jungle fowl into agricultural chickens, we were not initially selecting for like floppy ears or specific coat colors.
We were selecting for one singular overarching behavioral trait.
Tameness.
Exactly. Early humans needed animals that exhibited a reduced flight distance and a suppressed aggressive response.
We just wanted animals that wouldn't freak out and flee or try to bite us when we walked up to them. So you take the calmest wolves, you breed them together, and over generations you get a docile animal.
Simple enough on the surface.
Right. But the biology beneath that simple breeding program is astonishingly complex. Wrangham's hypothesis centers on a specific group of embryonic cells, neural crest cells.
Right, so neural crest cells are essentially a temporary group of highly migratory multipotent cells. They form very early in vertebrate embryonic development, right along the developing spinal cord.
And multipotent means they can turn into a bunch of different things.
Exactly. Because as the embryo matures, these cells migrate throughout the entire body to construct incredibly diverse systems.
Like what?
They differentiate to form sensory neurons, the cartilage of the face and ears, the melanocytes, which provide pigment to the skin and fur.
And critically, portions of the adrenal glands,
Yes, the adrenal glands.
which is the primary engine of the sympathetic nervous system. It's what pumps out the adrenaline and cortisol that drive an animal's fight or flight response.
Which connects directly back to human intervention. Wrangham hypothesized that when early humans selectively bred the tamest, least aggressive animals, they were inadvertently selecting for genetic mutations that resulted in mild neural crest deficits.
Oh, wow. So they were breeding animals that produced fewer migrating neural crest cells, or maybe cells that just migrated more slowly.
Exactly. And a reduction in neural crest cells reaching the adrenal glands means a smaller, less reactive adrenal system.
Which directly yields a tamer, less fearful animal.
Yes. But because those exact same neural crest cells are responsible for building so many other parts of the body, you get an unavoidable cascade of unintended physical side effects.
Which explains why domesticated animals all across the globe share those bizarre morphological similarities.
Right. If you reduce neural crest migration to get a tamer animal, you also reduce the cells that build ear cartilage.
Resulting in floppy ears.
You reduce the cells that build jawbones.
Leading to shorter muzzles and smaller teeth.
You reduce the melanocytes reaching the skin.
Resulting in white patches of fur, the piebald coloration. We never set out to breed dogs with floppy ears and piebald coats.
No, those traits just came along for the ride, entirely as a byproduct of selecting for docility.
The overarching genetic principle at work here is known as pleiotropy.
Pleiotropy, yes. It occurs when one single gene, or a tightly linked cluster of genes, influences two or more phenotypic traits that appear utterly unrelated on the surface.
We see this validated brilliantly in those top-down quantitative trait locus, or QTL, studies performed on the Bankiva chickens.
The wild ancestors of domestic chickens.
Yeah. The researchers mapped out the genetics of tameness. They identified a specific gene, a vasopressin receptor gene, where variations were strongly associated with low aggression. But because of pleiotropy, those exact same genetic markers completely altered the physical growth rates and even the comb size of the chickens.
It's all connected.
You know, the most intuitive analogy I can think of to explain pleiotropy, if you're trying to wrap your head around it, is the architecture of a massive software program.
Oh, the app update analogy. I like this one.
Yeah. Imagine an app developer releases an update designed strictly to fix a minor bug on the payment processing screen.
Okay.
However, because the underlying code of the app is this deeply interconnected web of shared libraries and dependencies, altering the code for the payment screen inadvertently changes the font size across the entire application and completely breaks the camera function.
The software update is pleiotropic.
Exactly. When human breeders tweaked the code for adrenal response, the ear cartilage code became compromised and the ears flopped over.
It perfectly captures the systemic integration of the genome. It reminds us that traits do not evolve in isolation.
Right. So when we observe an animal in the wild displaying a seemingly bizarre physical feature or a counterintuitive behavior, we really have to consider that it might just be a pleiotropic artifact.
A harmless byproduct of a highly advantageous trait being selected for in an entirely different biological system.
Okay, so returning to natural selection out in the wild. The sources explicitly lay out the three fundamental prerequisites that must exist for evolution to shape a behavior.
Right, the three rules.
First, there must be variation within the population, because if every individual behaves identically, there's nothing for the environment to select.
Makes sense.
Second, there must be fitness consequences. The behavioral variation has to actually yield a tangible difference in survival and reproduction.
And third, there must be a mechanism of inheritance. The behavior has to have a genetic basis so it can actually be transmitted to the offspring.
Right. And a classic framework for observing these three conditions in action involves measuring what researchers call approach scores in various bird species.
This is a standard behavioral assay. It's used to quantify an animal's reaction to novelty.
Neophobia, basically, or
Really?
boldness.
Right. So picture a population of birds encountering a completely novel object in their habitat.
Like a piece of brightly colored plastic that resembles a weird fruit, or an unfamiliar mechanical trap set by researchers.
Immediately, you observe the required variation. Some individuals in the flock are highly bold. They exhibit a low latency to approach. They fly right up to the strange object within 30 seconds to check it out.
What is this? Can I eat it?
Exactly. While other individuals are highly risk-averse, they maintain their distance, cautiously observing for 10 minutes before even making a move.
So there's the variation. And the fitness consequences of this variation become immediately apparent depending entirely on the state of the environment. If the novel object is actually a high-calorie food source, the bold bird capitalizes on the resource. It gains an energetic advantage and potentially increases its reproductive output.
While the cautious bird goes hungry.
Exactly. However, if that novel object happens to be a predator trap or a venomous insect it's never encountered.
The bold bird approaches quickly and is immediately removed from the gene pool.
Yep. The cautious bird survives. So depending strictly on whether the specific environment is food-rich or predator-heavy, the selective pressures will ruthlessly favor one behavioral phenotype over the other.
And because behavioral tendencies like boldness and neophobia are rooted in the animal's neuroendocrinology, meaning they have a mode of inheritance, the genetic makeup of the population will shift over generations.
Right. A forest filled with hidden dangers breeds a remarkably cautious population. A safe forest filled with ephemeral food rewards breeds a population of bold risk-takers.
It's an elegant conceptual framework.
It is, but it really demands that we look deeper. We understand that boldness is inherited, right? But how does a sequence of nucleotides, just sitting inertly inside a cell nucleus, actually translate into a bird actively choosing to fly toward a strange object?
We have to examine the physiological architecture of behavior.
Yeah. We have to bridge the gap from a microscopic gene to a macro-level physical action. So we have to look at the central dogma of molecular biology.
Right, because DNA does not directly compel an animal to run or fight or build a nest.
DNA serves purely as the instruction manual for the synthesis of proteins.
Yes. The DNA is transcribed into RNA, and the RNA is translated into proteins.
And those synthesized proteins then serve as the building blocks and the chemical messengers of the body. They construct the ion channels in the brain, they synthesize the neurotransmitters like dopamine and serotonin.
And they build the incredibly complex hormone receptor networks throughout the nervous system.
Right. It's the real-time functioning of that neurophysiological machinery that ultimately governs the animal's physical behavior.
And this construction process begins at the absolute genesis of the animal's life. When an embryo is nothing more than a microscopic cluster of a few dozen cells, those cells exist in a state that developmental biologists refer to as totipotent.
Right, to tipotency. It's such a remarkable biological state. It means that every single cell in that early cluster possesses the total genetic potential to differentiate into any specialized cell type required by the mature organism.
Be it a cardiac muscle cell, a retinal cell in the eye, or a motor neuron in the spinal cord, every single cell carries the exact same comprehensive genetic library.
Okay, but if they all have the exact same library, what determines their final identity? How does one become an eye and another become a heart?
The fate of the cell is determined entirely by gene expression, the highly coordinated process of turning specific genes on or off. This regulation is dictated by the chemical gradients and the cellular environment surrounding each individual cell as the embryo develops.
So the local environment basically forces a cell to read one specific chapter of the genetic manual and completely ignore the rest.
Precisely.
Which means the architecture of behavior is being built before the animal is even born. And the field of behavioral genetics provides some of the most compelling evidence for just how tightly behavior can be linked to DNA.
Oh, the Permiscus mice study.
Yes. The sources highlight this truly landmark study involving this specific genus of rodent, the deer mice.
The QTL analysis of the Permiscus mice is basically considered a masterclass in demonstrating the genetic basis of complex physical action.
It's unbelievable. So we're looking at two very closely related sister species. First, you have Permiscus maniculatus, the standard deer mouse. When it digs a burrow to live in, its architectural ambition is quite low.
It's a very basic builder.
Right. It digs a simple, short entrance tunnel straight down, creates a small nest cavity, and just stops.
Good enough.
Yeah. But then you have Permiscus polionotus, the old field mouse. This species behaves like a paranoid structural engineer.
It really does.
It digs a long, sweeping entrance tunnel, builds a nest cavity, and then actively digs a secondary escape tunnel that angles back up toward the surface of the Earth. But it stops just millimeters short of breaking through the topsoil.
The evolutionary logic behind that escape tunnel is brilliant. It's a dedicated anti-predator defense.
How does it work?
If a terrestrial predator, like a snake, infiltrates the main entrance tunnel, the mouse isn't trapped. It just pushes upward, bursts through that thin superficial layer of dirt at the end of the escape tunnel, and evades capture.
So clever. But the fundamental question for behavioral geneticists was whether this complex multi-stage construction project was a learned tradition-like passed down from mother to pup through observation, or if it was hardwired into their genetic code.
Right. And because maniculatus and polianotis are so evolutionarily close, researchers were able to bring them into the laboratory and successfully interbreed them.
Creating hybrid offspring.
Exactly. And creating hybrids allows researchers to observe how complex traits segregate or break apart in subsequent generations.
Right. By analyzing the DNA of these hybrids using that quantitative trait locus mapping we mentioned earlier, which basically correlates specific physical behaviors with specific genetic markers scattered across the chromosomes.
They achieved an astonishing level of resolution with this.
It completely redefines how we view the power of genetics. They discovered that the overall length of the Burrow system is controlled by exactly 3 specific genetic loci.
But far more incredibly.
Yeah. They found that the complex behavioral decision of whether or not to construct the secondary escape tunnel is dictated by just one single genetic locus.
One genetic switch.
It's mind-blowing. The precision is what makes the study so profound. We aren't talking about a generic gene for digging. We are looking at a highly specific sequential motor pattern.
The cognitive and physical act of digging an escape chamber.
Controlled by a singular switch. It provides indisputable proof that natural selection can ruthlessly fine-tune highly specific survival strategies by altering a remarkably small sequence of DNA.
But we should clarify, the influence of genes doesn't end once the animal's fully developed.
Right. It's not like the genes build the brain and then permanently clock out for the rest of the animal's life.
No, the neurobiology of decision-making relies on a genome that is highly dynamic and constantly responding to the animal's immediate environment.
The sources illustrate this beautifully with the acoustic communication of male zebra finches.
This is a perfect example of real-time phenotypic plasticity.
Right. So when a territorial male zebra finch hears the vocalized song of an unfamiliar foreign male of its own species encroaching on its territory, the auditory processing centers of its brain go into immediate overdrive.
And it's not just a cognitive realization that an intruder is present.
No.
The mere physical impact of those specific sound waves hitting the bird's ear causes an immediate molecular reaction. Within minutes, a specific gene located in the bird's brain, known as EGR1, is aggressively expressed.
And the EG1 gene operates as a transcription factor, right?
Yes. Its biological purpose is not to build a physical structure. It acts as a master switch that travels into the nucleus to turn a vast array of other downstream genes on or off.
So the acoustic signal from arrival physically rewires the neurogenomic state of the bird's brain in real time.
Exactly. This cascade of gene expression alters the bird's neurochemistry, which subsequently shifts its behavior. It primes it for heightened aggression, territorial defense, or vocal countersignaling.
Just from hearing a song. To fully map out these genetic pathways and isolate exactly what individual genes are responsible for, molecular biologists frequently employ a technique called genetic knockout engineering.
Typically utilizing laboratory mice.
Right. And the concept of a knockout mouse is straight out of science fiction. Scientists literally intervene at the embryonic stage and specifically knock out or permanently inactivate a targeted gene.
The mouse is then allowed to develop and mature without that specific piece of genetic code,
Allowing researchers to observe exactly what behavioral capacities are lost or altered.
and the unintended consequences of these targeted deletions reveal the hidden architecture of inhibition and drive in the mammalian brain.
Yeah, in one particularly striking experiment detailed in the literature, researchers targeted a gene that they believed was primarily associated with basic spatial learning and memory formation.
Okay.
But when the knockout mice matured, the researchers realized they had inadvertently created a population of super aggressive mutants.
Wow.
These mice exhibited an absolute absence of fear or social restraint. When they were introduced to other males, they engaged in physical combat so relentlessly and with such reckless violence that they would literally fight until they fractured their own spines.
The outcome is brutal,
It
but
is.
the scientific implication is vital.
Right.
It demonstrates that what we consider normal behavior, the capacity for fear, social submission, physical restraint, that is not simply the default state of the animal.
It's actively managed.
Continually managed by highly specific neurochemical pathways. When you genetically delete the pathway, the inhibition evaporates, leaving completely unchecked aggression.
And there's another knockout study that approaches behavioral genetics from the exact opposite emotional spectrum.
The maternal care one.
Yeah. Researchers deleted a single gene, and the resulting mutant mice appeared entirely 100% normal. They had standard metabolic rates, they navigated mazes flawlessly, they slept normally, and they engaged in standard courtship and mating behaviors.
But the instant the female mutants gave birth, the genetic deficit revealed itself.
They completely ignored their newborn pups. They simply walked away, entirely devoid of maternal instinct.
A single gene deletion completely severed the neurological reward circuit associated with pup retrieval and maternal care. It effectively wiped out millions of years of mammalian evolutionary programming.
It just underscores how fragile and deeply genetically dependent complex social behaviors truly are. But, this string of incredible examples, a single gene controlling an escape tunnel, an acoustic wave altering brain chemistry, a deleted gene erasing maternal care, it inevitably leads us to a bit of a philosophical cliff edge.
I know where you're going with this.
Right, because if our DNA exerts such profound surgical control over our actions, are animals merely meat robots? Are we looking at a system of strict genetic determinism where the environment is totally irrelevant and every movement is preordained by a sequence of nucleotides?
It's the most persistent and dangerous trap in behavioral biology, and the consensus of modern science vehemently rejects strict genetic determinism.
Thank goodness.
Genes are incredibly powerful, yes, but they do not operate in a vacuum. Genes provide the biological potential, physiological boundaries, the behavioral predispositions, but it is the environment that entirely dictates the ultimate outcome.
A colleague of mine once framed it with a beautiful analogy. Think of the genome as a highly complex musical score. The DNA provides the sheet music. The notes, the rests, the underlying melody, they're all permanently printed on the page. But the environment, the animal's life experiences, its nutritional status, the maternal care it receives, that is the orchestra playing the music.
I love that.
The orchestra ultimately decides the tempo, the volume, the emotional resonance, and whether certain passages are played loudly or entirely ignored.
That's a perfect metaphor.
Yeah.
And it serves as the ideal bridge into the reality of how behavior is actually manifested. Because to understand how the orchestra interprets the genetic sheet music, we have to move beyond the rigid sequence of DNA and explore the dynamic world of epigenetics, early development, and the profound impact of learning.
Exactly. Now, the historical approach to this topic was heavily bogged down by the nature versus nurture debate. We spent decades arguing over whether a behavior was purely innate, driven by nature, or purely learned, driven by nurture.
But modern behavioral ecology has definitively buried that dichotomy.
It's never nature versus nurture. It is an incredibly complex, inextricable tangle of both. And the most revolutionary mechanism illustrating this interaction is epigenetics.
Right. Epigenetics, which literally translates to above or on top of genetics, is the study of how environmental factors chemically modify the DNA structure to alter gene expression without ever changing the underlying nucleotide sequence of the genome itself.
So, returning to our musical analogy, epigenetics is the equivalent of a conductor taking a piece of heavy tape and sticking it over a specific key on the piano.
Exactly.
The physical key is still there, the note remains written in the sheet music, but the instrument physically cannot play it.
At a molecular level, this often takes the form of DNA methylation.
How does that work?
Environmental stimuli, say, a mother experiencing severe nutritional stress during pregnancy, or an animal growing up in a highly enriched, complex physical environment, trigger the attachment of chemical tags like methyl groups directly onto the DNA strand or the histone proteins that the DNA wraps around.
Okay, so physical tags.
Right, and these tags tighten the DNA coiling. They block the transcription machinery from accessing the genes, effectively silencing them. Alternatively, other epigenetic marks can loosen the DNA, massively boosting the expression of a gene.
And what makes epigenetics so paradigm shifting is that some of these environmental modifications can cross the generational barrier.
This is where it gets crazy.
Right. Certain epigenetic tags escape the cellular reprogramming that occurs during reproduction and are passed directly down to the offspring. We are looking at a mechanism of acquired inheritance that bears a striking, localized resemblance to Lamarckian evolution.
Yeah, if a male mouse is subjected to a severely stressful environment, his endocrine response alters the epigenetic markers within his reproductive cells.
And those stress-induced markers are transmitted through his sperm, permanently altering the baseline neurological development and stress reactivity of his pups before they are even born.
The environment is engaged in a direct chemical dialogue with the genome.
It's incredible. However, when we observe an animal adapting to its environment as it grows, we do have to be careful not to attribute all behavioral improvements to cognitive learning or epigenetic changes. We have to clearly distinguish between learning and physiological maturation.
Right, they are not the same thing.
It's highly intuitive to watch a young animal struggle with a physical task, slowly get better over a few weeks, and assume, it is practicing and therefore it is learning.
But that is frequently an illusion.
Yeah. The development of domestic chicks provides a fascinating case study here. When a newly hatched chick begins foraging, it instinctively pecks at small, contrasting objects on the ground that resemble seeds. However, a one-day-old chick has terrible aim.
It frequently strikes the dirt millimeters away from the target grain.
But over the subsequent few days, its pecking accuracy tightens up dramatically.
The immediate assumption is that the chick is acquiring hand-eye or beak-eye coordination through the trial and error of practice.
But elegant experimental designs have proven otherwise. Researchers have taken newly hatched chicks and fitted them with tiny prism goggles that artificially displace their visual field by a few degrees.
Just slightly off-center.
Right. So when the chick wearing goggles aims at a seed, the prisms ensure it pecks completely off target, hitting the empty ground. It never receives the physical reward of the seed.
Furthermore, researchers can simply withhold food and prevent the chicks from practicing the pecking motion altogether for the first few days of life, feeding them via alternative methods.
And in both scenarios, whether they're practicing with distorted vision and failing or not practicing at all, when normal vision is restored or the chicks are allowed to peck normally a few days later, their baseline accuracy has improved at the exact same rate as the control chicks who practiced normally.
So the improvement in targeting is driven primarily by the biological maturation of their visual cortex and cervical musculature.
Not by the cognitive acquisition of a skill.
Exactly. However, the chicks absolutely do learn what the appropriate targets are.
Right. Their physical capacity to execute a precise peck is a matter of maturation, but they rely heavily on experience to figure out that a yellow grain of corn is delicious, while a similarly sized yellow pebble is inedible.
Which brings us to specific modes of learning. Sometimes learning is not a gradual process of trial and error. Sometimes it's a rapid, localized neural event restricted to a highly specific biological window.
This phenomenon is known as imprinting.
The ethologist Konrad Lorenz made this concept famous through his work with gray-leg geese. He observed filial imprinting,
Which is the deeply ingrained process by which newly hatched precocial birds learn to recognize and follow their mother.
right, and Lorenz discovered that this learning does not occur flexibly throughout the bird's youth. It is confined to a critical sensitive period, lasting only a matter of hours immediately following hatching.
The underlying genetic programming dictates that the nervous system must lock onto the visual characteristics of the very first large moving object it encounters.
And in a natural ecological setting, that object is invariably the mother goose.
But the mechanism is blindly mechanical. In Lorenz's experiments, he ensured that he was the first moving object the goslings saw.
The gosling's neurobiology faithfully executed its programming and imprinted entirely upon a human scientist.
Or, in subsequent variations, upon a pair of rubber boots, or even a moving wooden box.
Crucially, once that narrow neuroplastic window closes, the biological concrete sets. The learning is virtually irreversible. The geese treated Lorenz as their parent for the rest of their lives.
It's an exceptionally rigid, biologically constrained form of learning. Most animals, however, rely on highly flexible learning mechanisms throughout their lives to adapt to fluctuating environments. These fall broadly into two categories: associative and non-associative learning.
Associative learning is what most people picture when they think of animal training as classical Pavlovian conditioning.
An animal learns to cognitively link two disparate stimuli.
Right, the dog learns that the sound of a bell reliably predicts the arrival of food, and soon the bell alone triggers salivation.
While non-associative learning is simpler, it involves altering a behavioral response based on repeated exposure to a single, isolated stimulus. Habituation is a classic example here.
A young animal might initially show a severe startle response to the shadow of a falling leaf. But after repeated exposure with no negative consequences, the nervous system habituates. The startle response is suppressed, and the animal saves its metabolic energy for real threats.
And the utility of individual experience is profoundly evident even in species that rely heavily on instinct. The sources detail the foraging behavior of the red-backed salamander.
Ah, yes.
Neonate red-backed salamanders are solitary from the moment they hatch. There is no parental care, no social mentoring. Within their first week of life, guided purely by instinct, they possess the complete neurological capacity to visually recognize, strike at, and capture small, evasive insect prey.
The baseline predatory sequence is entirely hardwired.
The behavioral ecologist Megan Gibbons and her colleagues sought to determine if these highly instinctive predators could actually improve upon their innate programming through individual experience.
And the data confirmed that they absolutely do. By repeatedly engaging in foraging behavior, the individual salamanders actively refine their technique.
They learn through trial and error which strike angles yield the highest capture probability,
How to physically handle struggling prey more efficiently.
and how to optimize their metabolic energy expenditure during a hunt. So the genetic instinct provides the fundamental framework, but individual cognitive learning polishes the behavior into a highly efficient survival tool.
Which brings us to one of the most intellectually thrilling and often misunderstood concepts in evolutionary biology.
I love this part.
If an animal can use cognitive learning to invent a new, highly advantageous behavioral trick during its lifetime, how does that newly learned behavior eventually become incorporated into the innate genetic instinct of the species thousands of years later?
Right, because we just discussed that Lamarckian evolution, the idea that a giraffe stretches its neck during its life and therefore has babies with longer necks, is fundamentally incorrect at the macro level.
Hmm.
A learned behavior doesn't directly rewrite the DNA sequence. So how do the genes know to adopt the learned behavior?
The solution to this paradox was proposed in 1895 by the psychologist James Mark Baldwin. It's known today as the Baldwin effect.
It outlines the precise mechanism by which behavioral plasticity, the capacity to learn, can actually accelerate and guide the trajectory of genetic evolution.
Let's break the Baldwin effect down meticulously because it is a vital concept for you to understand as a student. It operates as a distinct, multi-generational two-step process.
Okay, step one.
Step one begins with a population facing a novel environmental challenge. Imagine a sudden shift in climate introduces a new, highly nutritious but physically tough nut into a primate's habitat. The primates possess general behavioral plasticity. Through random trial and error, a few innovative individuals figure out how to place the nut on a flat stone and smash it with a heavier rock to access the calories.
This learned behavior provides a massive survival advantage.
Consequently, natural selection exerts intense pressure, heavily favoring any individual in the population that possesses the neurological capacity to learn this specific rock-smashing technique.
The generic ability to learn the task spreads rapidly through the population over subsequent generations.
Everyone is learning the trick, because if you don't learn it, you starve.
But relying on learning is incredibly dangerous, isn't it? It takes time, it burns metabolic energy, and a young animal might accidentally smash its hand or starve before it figures out the mechanics. Learning is a high-cost survival strategy.
Which sets the stage for step two, genetic assimilation. Because learning the behavior is both mandatory for survival and highly risky, natural selection will begin to intensely favor any random genetic mutations that make the acquisition of the rock-smashing behavior faster, more intuitive, or slightly more hardwired.
Oh, so if a mutation subtly alters the motor cortex to make a pounding motion more naturally rewarding, that individual survives more easily.
Exactly. Over thousands of generations, the genetic baseline steadily shifts. The selective filter constantly rewards the individuals who need the least amount of practice.
Eventually, what originated as a completely novel, cognitively demanding learned behavior becomes neurologically automated. It becomes genetically assimilated. The genes slowly catch up to the behavior that the animal pioneered through learning.
It is a profound realization. Through the Baldwin effect, the animals, by utilizing their cognitive flexibility to alter their relationship with the environment, effectively carve out the selective pressures that will shape their own evolutionary future.
And when we talk about the immense value and cost of learning, we must acknowledge the most efficient learning mechanism in the animal kingdom, social learning.
If learning through trial and error is risky, learning by observing the successes and failures of others is an evolutionary superpower.
Social learning drastically mitigates the lethal costs associated with environmental exploration. The most thoroughly documented and remarkable example of this phenomenon is the alarm call system of the vervet monkey. extensively studied by Dorothy Cheney and Robert Seaforth.
Right. When a predator approaches a group of vervet monkeys, they don't just emit a generic panic scream. They possess a highly sophisticated predator-specific semantic communication system.
It is a genuine vocabulary of danger.
The precision of the communication is tied directly to the necessary evasion tactic. If a vervet spots a leopard, it emits a specific acoustic sequence described as a bark. Leopards are cursorial, ground-based ambush predators. Therefore, the absolute correct life-saving response to a bark is to immediately abandon the ground and scramble high into the upper canopy of the trees.
However, if a vervet spots a martial eagle hunting from the sky, it emits a completely different vocalization, characterized as a short cough. Eagles attack the upper canopy from above. If a monkey hears the eagle cough and mistakenly executes the leopard response by running up to the top of a tree, it will be instantly killed.
The correct response to the eagle call is to drop out of the canopy and dive into dense, tangled bushes where the eagle cannot navigate.
And for a third distinct threat, such as a large python, the monkeys emit a chutter call. The response here is neither climbing nor hiding, but rather standing bipedally on their hind legs to aggressively visually scan the tall grass, effectively neutralizing the snake's reliance on stealth.
It is an incredibly complex system of semantic mapping. The critical question is, are these monkeys born genetically knowing this vocabulary, or do they have to go to vervet school?
The research on infant and juvenile vervets clearly demonstrates that while the biological capacity to vocalize is innate, the specific semantic application is entirely socially learned.
Initially, vervet pups are highly incompetent at the system. A pup might hear an adult give an eagle call, and instead of diving into a bush, the pup will simply sit paralyzed on a branch, looking around in total confusion.
Or worse, the pup will attempt to utilize the alarm calls inappropriately. Pups will frequently look up, spot a completely harmless falling leaf or a passing pigeon, and confidently belt out the eagle alarm call. Look out! A pigeon!
Right, and the learning process relies entirely on social feedback and observation. When a pup gives the eagle call for a pigeon, the surrounding adults immediately look up, visually assess the lack of threat, and entirely ignore the pup. There's no behavioral reinforcement.
However, if the pup correctly identifies a genuine predator and gives the accurate call, the adults react with violent, synchronized evasion. Through this constant localized social conditioning, the pups map the specific acoustic signals to the correct environmental triggers and the requisite physical responses.
Post-training analysis proves that pups raised in close proximity to experienced, vocal adults develop vastly faster reaction times and longer, more accurate scanning durations than pups deprived of experienced mentors.
This reliance on localized education is seen across taxa. We even see it in spatial navigation. Homing pigeons possess an incredible innate physiological tool magnetoreception. They can physically sense the Earth's magnetic fields to determine global direction.
But the sources note that a magnetic compass alone isn't enough to find a specific roost. Pigeons rely heavily on individual learning and social observation to map the specific olfactory gradients and the visual layout of the local landmarks surrounding their home loft.
The integration of innate biological tools with socially transmitted local knowledge is the hallmark of advanced ecological adaptation. But observing this vervet monkey system inevitably forces a broader question.
The culture debate.
Yes. If we have entire populations of animals transmitting complex, life-saving behavioral traditions across generations purely through social observation, saving their offspring from the deadly costs of trial and error, much like a human parent teaching a teenager how to navigate a busy highway, at what point does animal social learning graduate into the realm of culture?
You have touched upon one of the most fiercely debated topics in modern behavioral ecology. For decades, culture was defined as an exclusively human phenomenon. However, a growing consensus of researchers argues that when distinct behavioral traditions, whether they are tool use techniques in chimpanzees, specific vocal dialects in whale pods, or semantic alarm calls in monkeys, are reliably transmitted across multiple generations via social learning, entirely independent of genetic inheritance, we are undeniably observing the mechanics of animal culture.
The animals are building an informational legacy outside of their DNA. But regardless of how brilliant their culture or how refined their genetics, the ultimate unforgiving test of every behavioral adaptation occurs out in the wild.
It is time to shift our focus to the high-stakes ecological arena where these algorithms clash. The endless evolutionary arms race of eating and not being eaten.
In the natural world, predator and prey are locked in a relentless, synchronized evolutionary escalation. It is a biological manifestation of the Red Queen hypothesis. It takes all the running you can do just to stay in the same place.
If a prey species evolves a slightly more potent toxin or slightly better camouflage, the predator must immediately evolve a higher biochemical tolerance or sharper visual acuity, lest it face starvation and extinction.
We need to examine the offensive playbook first. The strategies predators use to secure calories. The simplest, though incredibly effective approach is the ambush predator, the sit-and-wait strategist.
Ambush predators rely almost entirely on absolute stealth, crypsis, and rapid acceleration. Consider orb-weaving spiders relying on nearly invisible tensile traps, or deep-sea anglerfish utilizing a bioluminescent lure dangling above massive jaws.
They minimize their own energy expenditure, remaining utterly motionless until the prey inadvertently wanders into the kill zone.
In stark contrast, you have the pursuit predators,
Hmm.
the active hunters who have evolved horrifyingly precise sensory superpowers to track their targets. The sources detail the astonishing capabilities of the Wells catfish.
The catfish uses its lateral line system, right?
Yes, the Wells catfish operates in deep, murky, or nocturnal aquatic environments where visual hunting is impossible. When a prey fish swims, the movement of its tail fin generates microscopic, swirling hydrodynamic wakes, essentially miniature underwater tornadoes.
And the catfish possesses specialized neuromast cells that can detect and analyze the specific pressure differentials of these invisible hydrodynamic footprints.
It allows the predator to accurately track a swimming prey fish over distance exceeding 50 of its own body lengths in complete darkness.
It is hunting by reading the turbulence of the water. That is incredible. But the predatory dynamic becomes exponentially more complex when individual hunters decide to pool their cognitive and physical resources. Cooperative hunting requires advanced social communication and strategic planning.
Group hunting significantly alters the ecological power dynamic, allowing smaller predators to reliably take down vastly larger prey and dramatically increasing overall strike success rates. The strategic approaches vary wildly.
Consider the lions of the African Serengeti. Group hunting here is almost exclusively the domain of the females. The lionesses fan out across the savanna, utilizing their tawny coloration to blend with the dry grass, executing a highly coordinated multi-flank ambush to trap a herd.
Utilizing stealth to close the distance before a rapid explosive sprint.
Contrast that stealth-based approach with the cursorial hunting strategies of African wild dogs or timber wolves. These are not primarily ambush predators, they are endurance athletes. Their strategy is fundamentally psychological and cardiovascular.
They hunt by deliberately charging a massive herd of wildebeest or caribou, aggressively inducing panic.
They want the herd to run because running reveals the hidden data. As the herd panics, the pack meticulously evaluates the locomotion of the individuals. They are looking for subtle signs of weakness, an older animal, a juvenile, or an individual masking a slight injury.
Once the pack identifies the optimal target, they coordinate their positioning to systematically separate that specific animal from the protective bulk of the herd.
They then engage in a prolonged pursuit, with individual pack members taking turns leading the chase to maintain immense pressure while conserving their own energy until the prey physically collapses from cardiovascular exhaustion.
If you are the prey animal residing in this terrifying landscape, how do you mathematically survive against thermal tracking, hydrodynamic sensors, and coordinated pack hunting? What does the defensive playbook look like?
The most metabolically efficient and primary defense mechanism is avoiding detection entirely. Crypsis, or camouflage. The evolutionary power of crypsis is beautifully illustrated by the classic research of Francis Sumner on the beach mice.
Sumner mapped out how the physical environment directly edits the genome of the prey. The populations of these mice that reside on the bright white sandy beaches of the coastal islands have evolved remarkably pale fur.
They blend in perfectly with the sand, rendering them effectively invisible to highly visual aerial predators like hawks and owls.
But if you travel just a few miles inland to populations of the exact same species living on dark, loamy soil, the mice possess deep, dark brown fur.
It is a perfect demonstration of localized natural selection. Any mouse carrying the genetic allele for dark fur on the white beach was rapidly consumed, selectively removing that specific genetic sequence from the coastal population.
But camouflage is a passive defense, and it inevitably fails. When the predator breaches the stealth perimeter, the prey must transition to active evasion. If the prey is physically faster, the biological imperative is simple: flee.
But what if the prey lacks the locomotive architecture for speed? What if you are a slow, metabolically sluggish caterpillar?
When speed is not an option, the The prey must deploy a portfolio of aggressive, chemically noxious, or radically evasive behaviors. When a caterpillar is physically contacted by a parasitoid wasp or a predatory bird, it will violently and erratically thrash its body to break the predator's grip.
It may abruptly regurgitate highly toxic or unpalatable fluids synthesized from its diet. Or, as a last resort, it will simply release its grip on the leaf and execute a freefall drop from the canopy into the leaf litter below.
Calculating that the risk of the blind fall is statistically preferable to the certainty of the bird's beak.
Beyond immediate evasion, some prey animals possess the astonishing ability to structurally rebuild their own bodies in anticipation of a predator. This phenomenon is known as inducible defenses or phenotypic plasticity.
Inducible defenses represent the ultimate triumph of environmental responsiveness. A remarkable example is found in the brown anole, a highly adaptable species of lizard. When terrestrial predators, such as larger ground-hunting lizards, are newly introduced into the brown anole's ecosystem, the anoles do not simply suffer higher mortality.
Their first response is behavioral. They immediately shift their spatial habitat, moving higher into the arboreal canopy to avoid the ground threat.
But the adaptation doesn't stop at behavior. The sources show that as subsequent generations of anoles grow and mature entirely in the trees, their physical morphology alters. They literally grow shorter legs.
Shorter limbs lower their center of gravity, providing vastly superior balance and gripping efficiency on thin, unstable arboreal twigs compared to the long legs required for ground sprinting.
Another profound example of inducible morphology occurs in the aquatic environment with frog tadpoles. If a tadpole is developing in an isolated pond devoid of predators, it grows a standard, metabolically efficient tail.
However, if that same pond contains predatory dragonfly larvae, the tadpoles can chemically detect the chiromones, the specific chemical signatures excreted by the predators.
The mere chemical presence of the predator triggers a developmental shift, causing the tadpole to invest massive biological resources into growing its significantly deeper, more muscular tail fin, vastly increasing its burst swimming speed to escape the dragonflies. The physical environment literally sculpts the anatomy.
Those are physical changes. But the most intellectually counterintuitive interactions in the entire predator-prey arms race occur when prey animals actively, deliberately interact and communicate with the predators trying to kill them.
The behaviors associated with pursuit deterrence hypothesis frequently baffle observers.
It completely upends our basic assumptions about self-preservation. If a gazelle spots a cheetah in the grass, every ounce of human logic dictates that the gazelle should freeze to utilize its camouflage or immediately run in the opposite direction. You do not advertise your location.
And yet, many prey species go out of their way to make themselves agonizingly conspicuous. African impalas perform a behavior called stotting, where they bound dramatically, feet off the ground, high into the air with stiff legs.
Various species of deer perform tail flagging, lifting their tails to expose a brilliant flash of bright white fur as they run.
To the untrained eye, it appears the prey is openly mocking the predator, shouting, "Here I am, come and eat me." But through the lens of behavioral ecology and signaling theory, the message is the exact opposite. It is a highly calculated transmission of data.
Because almost all predators, especially solitary cats, rely heavily on the element of surprise to minimize the massive energetic cost and physical risk of a chase.
Precisely. When an impala stots upon spotting a lion, it is executing an honest signal of physical fitness. It is communicating multiple pieces of vital information to the predator. First, I see you. Your stealth approach has entirely failed. You have no element of surprise. Second, observe the immense kinetic energy of my jump. I am in peak physical condition. I am not sick, I am not injured, and I have vast reserves of stamina. If you choose to initiate a pursuit, you will expend a catastrophic amount of metabolic energy, and statistically you will fail to catch me. Abandon the hunt and seek a weaker, unaware target.
It is a calculated behavioral bluff that perfectly manipulates the predator's internal energy calculus. And the data shows it works. The predator receives the signal, evaluates the ruined stealth and the obvious fitness of the prey, and frequently abandons the hunt without taking a single step.
The hypothesis is further supported by the fact that animals strictly modulate this behavior based on proximity. If the lion is 100 yards away, the impala stots. If the lion manages to creep within 10 yards before being detected, the impala abandons the signaling entirely and just scrambles for its life. The safety buffer is gone.
While stotting is an honest signal of high fitness, prey animals are equally adept at utilizing pure deceptive signaling. The broken wing display of the ground-nesting plover is a masterpiece of cognitive manipulation.
If a terrestrial predator, like a coyote or a fox, wanders too close to a plover's nest full of highly vulnerable chicks, the mother bird does not attack the vastly larger predator, nor does she hide. She initiates a highly choreographed theatrical performance.
She runs directly toward the predator's line of sight, collapses on the ground, drops one wing at an agonizing angle as if the bone is entirely shattered, and begins to desperately limp away from the location of the nest.
She is weaponizing the predator's instinct to seek the easiest possible meal. She is deliberately presenting herself as highly vulnerable, injured, and calorie-dense prey.
The coyote locks onto the seemingly injured bird and begins to follow. The mother plover carefully regulates her speed, limping just fast enough to stay out of the predator's jaws, meticulously luring the coyote further and further away from the hidden nest.
Once she calculates that the predator has been drawn a safe distance from her chicks, the theatrical performance concludes. The shattered wing magically heals and she launches into the air, flying away to safety, leaving the predator utterly bewildered and the nest entirely secure.
It is a brilliant exploitation of predator psychology. Another extreme manipulation of predator neurobiology is tonic immobility, colloquially known as feigning death. If a domestic hen is actively chased and physically cornered by a predatory cat, the hen will suddenly collapse and go completely rigidly paralyzed.
It looks exactly as if the bird has died of sheer terror, but it is a neurological hack. The predatory kill sequence in many felines and canines is deeply tied to the visual processing of movement and the physical sensation of struggle.
When the prey goes entirely rigid, the sensory inputs that drive the killing bite are suddenly silenced. The predator's aggressive sequence is interrupted. It becomes confused, often relaxing its grip or dropping the prey to investigate. In that brief window of predator confusion, the hen's paralysis breaks and it violently escapes.
The researcher Jeffrey Masson extensively documented these profound psychological interactions. Predators are not mindless killing machines. They are highly sophisticated risk management computers, constantly scanning for microscopic tells. Manson detailed observations of spotted hyenas that perfectly illustrate this terrifying perceptual acuity.
Researchers in the field would frequently utilize a tranquilizer dart to briefly sedate a wildebeest for routine tagging and blood work. After the procedure, the animal would wake up, the sedative would wear off, and the wildebeest would rejoin the herd. To the human biologists observing through binoculars, the previously tranquilized animal looked absolutely 100% normal. It grazed normally, it stood with the herd, it ran at the exact same speed as the others.
But the visual processing of the hyenas operated on an entirely different level of resolution. The hyenas immediately recognized the herd, bypassed the truly healthy animals, and zeroed in with lethal precision on the specific individuals that had been recently tranquilized.
The hyenas were detecting some infinitesimal, almost invisible alteration in the animal's gait, a microscopic delay in its reaction time, or a subtle postural droop, a micro-expression of vulnerability that human eyes were biologically incapable of seeing.
So when we look at the totality of this arms race, we realize that the prey animals are effectively sitting at a high-stakes poker table with the predators. They're constantly managing risk by utilizing bluffs, like the plover's broken wing, and deploying deliberate tells like the impala's tail flagging to manipulate the predator's psychological calculus.
That is an excellent summation of the individual struggle for survival. However, while avoiding the jaws of a predator is largely a game of individual self-preservation, many species across the evolutionary tree have pioneered a fundamentally different, vastly more complex, and deeply counterintuitive survival strategy. They rely entirely on the social group, often to the point of absolute lethal self-sacrifice.
Which brings us to the final, most philosophically dense topic of our deep dive: the profound paradox of society, altruism, and the concept of the selfish gene.
Before we dissect the mechanics, we must strictly define our terms. In standard human conversation, altruism implies a moral goodness or charity. But in evolutionary biology, altruism is defined by a rigid mathematical equation. An altruistic behavior is any action that actively increases the reproductive fitness of a recipient individual while simultaneously decreasing the direct reproductive fitness of the actor performing the behavior.
You do something that helps another animal survive and reproduce, but it costs you precious calories, time, mating opportunities, or in the most extreme cases, your own life.
And this specific definition posed a massive existential threat to Charles Darwin's entire theoretical framework. If the fundamental engine of natural selection ruthlessly favors physical traits and behaviors that maximize an individual's own direct reproductive success, how could a behavior that actively harms or kills the individual ever evolve? The selfish gene theory, popularized by Richard Dawkins, dictates that genes exist solely to replicate themselves. Altruism appears to be a blatant violation of the core rules of biology.
But the natural world is overflowing with altruistic acts. A Belding's ground squirrel spots a predatory hawk, and instead of silently slipping into its burrow to save itself, it stands upright and emits a piercing alarm call. That call alerts the rest of the colony to hide, but it acts as an acoustic beacon, drawing the hawk's lethal attention directly to the caller. Why does the squirrel commit evolutionary suicide? The key to unlocking this paradox lies in the brilliant mathematical framework of kin selection.
The conceptual breakthrough for kin selection was perfectly encapsulated by the evolutionary biologist J.B.S. Haldane, who famously quipped while sitting in a pub, "I would gladly lay down my life for 2 brothers or 8 cousins." This seemingly humorous statement is the literal mathematical engine of altruism.
Because you do not hold a monopoly on your own DNA. You share a massive percentage of your genetic code with your relatives.
Precisely. You share exactly 50% of your genetic alleles with a full sibling, 25% with a nephew or an aunt, and 12.5% with a first cousin. Evolutionary fitness is not merely about how many children you personally produce. It is about inclusive fitness, the total number of your genes that make it into the next generation, regardless of whose body they reside in.
Therefore, if you sacrifice your own life to save the lives of 3 of your full siblings, you lose your 100% genetic representation, but you ensure the survival of 150% of your identical genetic code distributed across your siblings. The cold biological math works out. In studies of complex primate societies, behavioral ecologists have calculated the precise thresholds for these actions. They use an R value, the coefficient of relationship. In many primate groups, an R value of 0.125, the genetic equivalent of a first cousin, represents the critical biological threshold. If the relationship falls below that number, true, risky altruism effectively drops off a cliff.
The absolute zenith of kin-selected altruism is found in the eusocial insects. The complex societies of bees, ants, and termites are marvels of genetic coordination.
In a honeybee hive, you have tens of thousands of female worker bees who are entirely sterile. They will spend their short, frantic lives foraging for nectar, performing incredibly complex vector calculus waggle dances to communicate the exact coordinates of food sources to their sisters, and violently defending the hive with a stinger that rips their own abdomen apart when deployed. They possess zero chance of ever reproducing directly.
Similarly, in termite colonies, you have specialized soldier castes with massive, heavily armored heads. When the colony is breached by invading army ants, these soldiers will rush to the fracture point and literally wedge their own heads into the tunnels, allowing the workers behind them to seal the breach with mud, permanently entombing the soldiers outside. They willingly sacrifice themselves because the unique genetic architecture of these colonies ensures they share an extraordinarily high percentage of their genome with their siblings. By dying to protect the reproductive queen, they are securing the transmission of their own genetic legacy with far greater efficiency than if they reproduced independently.
Kin selection neatly explains why relatives help each other. But the biological world presents us with another massive puzzle. How do we explain complex cooperation and altruism between animals that are completely genetically unrelated?
The evolutionary mechanism that governs cooperation among non-relatives is known as reciprocal altruism. In colloquial terms, it is the biological implementation of the phrase, "I will scratch your back today if you guarantee you will scratch mine tomorrow."
But reciprocal altruism is cognitively demanding. Unlike kin selection, which can operate purely on chemical scent recognition, reciprocal altruism requires significant brain power. You have to be able to individually recognize the members of your group, you must possess the memory capacity to recall past interactions, and you must have the cognitive architecture to detect and punish cheaters, individuals who accept help but refuse to return the favor.
Which is why we observe reciprocal altruism primarily in highly intelligent, long-lived social species like bats, cetaceans, and primates. Let's return to the vervet monkeys for a brilliant experimental demonstration of this concept.
Researchers wanted to prove that monkeys actively track favors. They set up an acoustic playback experiment in the wild. They hid high-quality speakers in the dense bushes surrounding a troop of vervets and played the pre-recorded, highly specific distress calls of a specific individual monkey soliciting physical help. Essentially, playing a recording of monkey A screaming, "Help! I am being attacked by a rival."
The researchers meticulously observed the reaction of the rest of the troop to this phantom distress call. The data revealed a stark, mathematically precise social ledger. They found that an unrelated monkey was vastly more likely to stop foraging, run toward the speaker, and risk entering a physical altercation to help the calling monkey only if those two specific individuals had engaged in a prolonged mutual grooming session earlier that same day. If they had not recently engaged in a cooperative exchange, the distress call was largely ignored.
The monkeys are quite literally maintaining a mental microeconomy. You invested 20 minutes picking parasitic ticks out of my fur yesterday, depositing social capital into my account. Therefore, I am willing to expend energy and risk physical injury to assist you in a fight today.
It is a highly calculated, delayed exchange of biological favors, but we must be careful not to romanticize social living. The benefits of cooperation are immense, but social groups are also cauldrons of intense, localized competition. There is a dark side to society, constant conflict over limited resources.
Right. If you live in a dense group, you're constantly shoulder to shoulder with your greatest rivals. You're competing every single day for the safest sleeping site, the most calorie-dense food, and the most fertile mates.
If that competition resulted in physical combat every single time two animals wanted the same piece of fruit, the society would collapse due to injury and energetic depletion. To mitigate this constant lethal friction, social animals establish rigid dominance hierarchies, commonly referred to as pecking orders. Once the initial physical conflicts determine the hierarchy, the social structure stabilizes. Subordinate individuals instinctively defer to the dominant alphas regarding resources without initiating a physical fight, which saves massive amounts of metabolic energy and prevents catastrophic injuries for the entire group.
From a human perspective, we naturally assume that being the alpha, the dominant individual at the absolute pinnacle of the hierarchy, would represent the ultimate stress-free existence. You go first access to all the food, you monopolize the mating opportunities, everyone defers to you. It seems like the biological jackpot.
It is a highly logical assumption, but biological reality frequently subverts our expectations. A landmark 2011 study published by Gesquier and a massive team of researchers shattered this assumption by measuring the actual physiological toll of dominance in wild baboon troops in Kenya.
The researchers tracked the baboons for years, meticulously collecting fecal samples to measure the exact levels of glucocorticoids, the primary stress hormones, like cortisol, circulating in the animals' bloodstreams.
The prevailing hypothesis was that the subordinate males, constantly bullied and deprived of resources, would exhibit the highest stress levels. The data revealed a shocking U-shaped curve. In a stable social hierarchy, the subordinate males were indeed highly stressed. But astonishingly, the most dominant individuals, the alpha males, exhibited stress hormone levels that were just as high and frequently higher than the lowest-ranking subordinates. It was the beta males, the middle management of the troop, who experienced the lowest stress.
How does that make evolutionary sense? Why is the king the most stressed animal in the troop?
Because heavy is the head that wears the biological crown. The alpha position is not a state of relaxation. It is a state of perpetual, exhausting hypervigilance. The alpha male must constantly aggressively fend off physical challenges from ambitious betas. He must spend immense amounts of metabolic energy mate-girding the fertile females to ensure exclusive reproductive access. He must aggressively mediate disputes among subordinates to maintain troop cohesion. It is an incredibly energetically demanding and psychologically toxic position to maintain. The alpha burns out quickly.
That research introduces a crucial word: psychology. When we discuss kin selection math and reciprocal ledgers, it sounds incredibly cold, but the sources actively delve into the genuine emotional lives of animals. The mathematics of evolution produces the raw, visceral emotions the animals experience.
We cannot separate the evolutionary logic from the physiological sensation. Consider the phenomenon of surplus killing. We observe a weasel infiltrating a dense henhouse and methodically killing every single bird in the enclosure, executing dozens of chickens when it can only physically consume a single breast. Or a pack of hyenas slaughtering an entire herd of gazelles trapped in a box canyon. We view this as malicious or wasteful, but biologically it is a neurological misfire. The predatory drive, the sheer dopamine rush and neurological reward of executing the kill sequence is so deeply, powerfully ingrained in the predator's brain that when it is suddenly presented with an artificial, hyperdense prey environment where escape is impossible, the satiation off switch fails to trigger. They hunt repeatedly because the physiological act of hunting feels incredibly overwhelmingly good.
And on the exact opposite side of that behavioral spectrum, we see that same physiological reward system driving the evolution of empathy and social bonding.
Precisely. We look at neurohormones like oxytocin and vasopressin. Millions of years ago, in primitive mammals, these chemical circuits evolved for a highly specific, localized purpose to regulate sexual behavior, uterine contractions, and maternal milk letdown. They rewarded the basic act of reproduction.
But evolutionary opportunism strikes again.
Yes, over millions of years of mammalian evolution, those ancient, powerful hormonal circuits were co-opted, expanded, and repurposed. The neurological reward system that initially just made a mother feel intensely bonded to her immediate nursing infant was slowly broadened to make animals feel a deep sense of reward and lowered anxiety when in close physical proximity to their extended family and eventually their broader social group. This hijacked hormonal circuitry is the literal physiological root of social attachment, empathy, and the psychological drive to perform altruistic acts. Helping a friend releases a chemical reward in the brain.
Which brings us to the ultimate philosophical synthesis of the selfish gene concept. If every single thing an animal does, from a bird singing a territorial song, to a monkey grooming a rival, to a human experiencing the profound warmth of empathy in helping a neighbor, is fundamentally, biologically driven by an underlying evolutionary desire to ensure the survival and replication of our DNA, isn't all altruism at its deepest core just genetic selfishness?
From the strict molecular perspective of the gene itself, yes, the gene only knows replication.
It makes me think of a multi-generational family business. Imagine you are the CEO. You might take a massive, painful pay cut during an economic downturn. You might work exhausting 80-hour weeks, and you might completely drain your own personal savings account to ensure the family company survives a bad year. To the outside observer, your actions look incredibly noble, selfless, and purely altruistic. You are sacrificing your own comfort. But ultimately, at the core of the decision, you're doing it because you want your family's overall legacy, the accumulated wealth, and the family name to be protected and passed down to your children. The genes are the family business. Every seemingly selfless act is an investment in the long-term survival of the biological portfolio.
It is an exceptionally pragmatic and perhaps slightly cynical way of viewing the beauty of the natural world, but from the standpoint of evolutionary behavioral ecology, it is entirely accurate.
We have covered a truly immense amount of intellectual ground today. We set out to provide you with a comprehensive, foundational understanding of behavioral biology. And the overarching narrative we have uncovered is that animal behavior is never magic. It is never random.
It is a deeply logical, profoundly elegant, and beautifully complex web of biological interactions. We started by seeing how natural selection acts as the relentless engine of design, tinkering with ancient DNA, shaping everything from the tameness of a domesticated dog through the migration of embryonic neural crest cells to dictating the exact genetically coded length of a deer mouse's escape tunnel. We saw how the dynamic physical environment plays the static genetic sequence like an orchestra through the chemical tags of epigenetics. We explored how cognitive learning from a tiny salamander refining its strike angle to a vervet monkey mastering the semantic vocabulary of predators fundamentally alters the trajectory of a species' evolution.
We examined the brutal high-stakes arms race of predators and prey, the psychological warfare of bluffs and deceptive signals, and finally, the incredible cold evolutionary mathematics that drive a termite to willingly sacrifice its life for the colony, or a baboon to endure the toxic stress of the alpha crown.
So here is how you take this academic knowledge and apply it to the real world. The next time you step outside, take a moment to truly observe. When you see a simple bird at a feeder making a split-second decision to flee, or you watch a domestic dog flawlessly interpret a human hand signal, or you notice a squirrel freeze rigidly when the shadow of a cloud passes overhead, do not just see a cute animal. Look deeper into the machinery. You are witnessing hundreds of millions of years of ruthless natural selection, a highly tuned dynamic nervous system, generations of epigenetic history, and real-time environmental feedback, all converging and executing simultaneously.
You are watching the algorithms of life successfully compiling and running their code in the wild.
And that leaves me with one final, deeply provocative thought for you to chew on before you return to your studies. We spent significant time unpacking the Baldwin effect, the proven theory that a cognitively learned behavior, if it becomes absolutely crucial for survival, can effectively guide natural selection and eventually become permanently hardwired into the DNA of the species over thousands of generations. And we discussed the reality of epigenetics, how the immediate physical and psychological environment can physically attach chemical tags to our genome that alter gene expression in our children.
Both are powerful, proven biological mechanisms by which external experience physically reshapes internal genetics.
Exactly. So, turn that lens away from the animals and look closely at humans. Look at our current unprecedented global culture. Look at our total reliance on smartphones for navigation and memory, our constant exposure to artificial blue light, our highly processed diets, and our perpetual state of digital hypervigilance and social media stress. If the Baldwin effect is actively operating and epigenetics is constantly tagging our DNA in response to stress, are human cultures and modern technology actively, right now, rewriting our own genetic code? Will the cognitively learned behaviors of navigating a digital world and the physiological adaptations to artificial light and constant digital stimulation eventually become permanently hardwired into the human genome 1,000 generations from now?
Hmm.
Are we blindly domesticating ourselves into a new species? Think about the biological legacy of that the next time you stare at a screen.
It is a conceptually fascinating and genuinely terrifying evolutionary prospect.
It brings us right back to our starting point. The diagnostic machine of biology is entirely broken, the engine is constantly running with the hood open, and the system is actively rewriting its own source code as it hurtles forward. Thank you for taking this deep dive with us. Keep questioning the world around you, keep observing the hidden algorithms, and we will see you next time.